I guess I'm hot wired. Hi, everybody. Well, I'm kind of glad some people are going to show up. I was afraid I was going to have to talk to the wall back here. There's a second level in that the church doesn't have PowerPoint, so I don't know if this is going to work or not. We'll see. We'll kind of wing it here for most of the things. So what I'd like to talk about today are some things that have to do with a research arm from my particular interest that has to do with gastroenterology and gastrointestinal pathophysiology. And I'll talk a little bit here about gut microbiome, what that means, and how that can impact individuals in terms of overall health, and not just physical health, but also mental health, mental well-being. Your clicker is not hot. Okay, this is what I mean, that PowerPoint is going to be corrupted in some cases, so maybe we'll have to kind of work through this here. So what we're going to talk about here is actually a two-way interaction. There is, I think in the kind of general public, there's a misconception that most gut microbiome phenomena is a sort of bottom-up phenomenon, but actually there's a top-down control, so that there's a two-way street. And I've tried to emphasize this in this figure, that there's a two-way arrow here, that there's a communication, a two-way street. And in fact, there's a triangulation. So at least the bottom portion of the slide is showing up, that there's all our organ systems, our physiology, our overall health is actually a triangulation with our brain. And what is hidden behind here anyway is our gut, which contains most of the microbiome in our body. So what we're going to be talking about throughout this whole session, the triangulation of the gut and the brain and the rest of our body in a communication, an interactive communication. And the topics that I'll be covering so that we can put this all together is, first of all, I'm going to introduce this concept of people being meta-organisms. That is to say that we're a physiological conglomerate of intertwined people cells, human cells, and bacterial cells. Secondly, one of the topics I'd like to cover is a niche in microbiome, and that is COVID, believe it or not. There is a gut receptor. In fact, the gut is the single largest receptor surface area in the body for the coronavirus that causes COVID-19. And I'll give you a little bit of a role on how this is part of our microbiome, the COVID virus, in addition to bacteria that interact with the COVID virus, which causes some inflammation. And I'll kind of get into the mechanism of that. And then, well, here, once again, our two-way arrow is missing. And then what I'm hopefully putting this all together is the concept here that the gut microbiome interacts with the brain and the rest of the body to form a physiological situation with our nervous system and our metabolism that can control blood pressure. Gut microbiome plays a role in mood states, and I'll talk a little bit about depression and anxiety, and has an overall governing effect on our health, just our general overall health. Oh, there's my arrow. Thank you for introducing at least the arrow. So the premise here is that the gut microbiome plays an intimate role in human physiology. And the reason why this plays such an intimate role is that through evolution, we have co-evolved so that our bodies, your body, contains half people cells and half bacterial cells. You are comprised of 50% of the cells in your body are bacterial cells, microorganisms. There's about 100 trillion of your own cells and about the same amount, 100 trillion of bacterial cells. And a concept that I published some articles on this, and I'm trying to coin this phrase here, is that people are meta-organisms of this conglomerate. And in terms of overall health, we are an interactive, intertwined communication of people cells, bacterial cells, a crosstalk two-way direction so that this controls our overall physiology and our bacterial physiology. It controls our levels of inflammation, controls immune system events through regulation of bone marrow and spleen physiology. We have a role in our digestion and absorption of nutrients in the gastrointestinal tract. Neural circuits are involved, including cognition and mood circuits. The kidney is affected by this, including sodium and water and fluid and electrolyte balances, and our cardiovascular system is involved. And so you might think, well, you know, if we're a meta-organism, what role do bacteria play in some of these things? And I'm going to put this together for you, and hopefully at the end, if we'll talk through the slide anyway, you'll see the assemblage of this, how people are meta-organisms comprised of half bugs and half people cells. To do this, I want to show you how we come to this point in time through evolution to form ourselves as a kind of evolutionary adaptive entity called a meta-organism. And I'll kind of pick up the story in our evolution in the let's say 30,000 years ago in the hunter-gatherer phase of human evolution, and take you through kind of where you're sitting here in the chair in modern times. So as you know, we have evolved in a social order so that we're kind of tribally oriented. And our bodies are set up this way, our minds are wired this way. And in fact, see this guy, he's wondering whether his tribe wants to watch MSNBC or Fox News, okay? And not only do we have tribal conglomerations, but we do this to preserve resources and to protect ourselves from environmental assaults. Not only predators and environmental assaults in terms of climate and that sort of thing, but also unseen threats through pathogens, bacteria, microorganisms that could kill off an animal. And so what we have seen here is that these conglomerates, these aggregations of people kind of keep certain bacteria in their tribe. And through evolution, through evolutionary pressure and the adaptation that occurs in evolution, we've taken on good guy bacteria, so-called commensal bacteria, that can set up a chemical warfare with bacterial pathogens. And we have co-evolved with these bacteria. They get something from us, we get something from them. And so through the evolutionary pressure of the necessity of good guy bacteria maintaining a housing situation in our bodies, we have actually co-evolved to be a meta-organism, a conglomerate of bacteria and people cells that are inextricably linked together. And if we move forward in time, what happens is, since we're an animal with higher central nervous system wiring, the bacteria and our own physiology have set up a system so that the evolutionary pressure favors us to be in a slightly inflammatory biased situation. And the reason is twofold. First of all, it controls our brain to keep us out of trouble. So part of our central nervous system activity in our brain is kind of alarm systems to be vigilant of our environment and avoid things that get us into trouble, and in fact avoid us to kind of stop the propagation of that particular tribe, stay out of trouble. And our bacteria keep us in a state so our brain sort of is in this vigilant state. Not only that, but our whole physiology is set up so that if we encounter damage to our bodies, that the inflammation state will help in wound healing, help fight off bad guy bacteria through infections. And it's the commensal bacteria that keeps us in this particular state. So there's a kind of mutualistic benefit that you're sitting here in the chair today, reaping the benefits of our co-evolution with bacteria, so that there is a microbiome, gut, brain, organ interaction in an intertwined crosstalk. And our central nervous system takes advantage of this and has been steered in the direction of our particular behavior and cognitive capabilities. And in all of biology, there's a use it or lose it phenomenon in terms of evolution. And so there's actually adaptations for our bodies and our brains for developing depression, anhedonia, or kind of lack of interest, lack of curiosity, withdrawal, and the so-called sequelae of physiological and mental phenomena known as sickness behavior, flu-like symptoms, are all very much related to keep us out of trouble. And the problem is that there's no free lunch in the universe, and that there is some collateral damage that comes from keeping us in this inflammatory state. And that is, some organs kind of get a hit from that, and some organ systems and overall physiological entities like blood pressure can take a hit from the fact that even though we're keeping ourselves out of trouble, it's at the expense of perhaps a ding on some of the biology that is makes us human beings as kind of biological entities. Let me turn our attention to COVID-19, because this is a part of the gut microbiome, and I'll kind of show you how this plays a role. In early 2020, there was a paper published in Science. This is a prestigious research magazine publication, and the title of that paper was that the SARS-CoV-2 virus, which is the coronavirus that causes COVID, is bound to a protein called ACE2, angiotensin converting enzyme type 2. And this I saw this paper and it just like blew my socks off. I could not believe this, as that this particular paper came out. And if you look at the abstract here, the reason this blew my socks off is because it says here that ACE2, in the presence of an amino acid transporter, an intestinal amino acid nutrient transporter, forms a complex, and I'm not going to go into what that complex is, that is involved with coronavirus entry into our bodies. I thought, holy cow, this is unbelievable in 2020. And the reason I was so stunned by this is because I happen to remember that there was a paper published in 2010, okay, 10 years earlier. There was a paper published, and I'm going to zoom in on the text here for you. And the paper said, the author of the paper said, not a paper, this is a book chapter, okay? This book chapter says, our laboratory demonstrated the intestine has ACE, angiotensin converting enzyme, in it, and that this, combined with observations in the literature, was that it also has ACE2, and that during the 2003 through 2005, remember there was that MERS and SARS and CoV outbreak? Okay, so this paper was putting this together in 2010, and it said, if the body has ACE2 and it's bound to this amino acid transporter, this is an amino acid nutrient transporter, what does that have to do with ACE2? But these two concepts together, it says here, is predicting that perhaps the amino acid transporter with ACE2 plays a role in allowing this kind of a group of viruses called coronaviruses, the original CoV, not CoV2, but the original CoV, was playing a role in getting coronaviruses into our body. And so I was thinking, wow, this paper, or this book was published in 2020, 2010, 10 years earlier. What a prescient observation. And the reason why I was so stunned and remembered who this author was is because I wrote that book. I was flabbergasted that finally, you do these things and you put them in a dusty shelf and nobody cares for years. And then it finally kind of comes around and is kind of shedding some light on some of the work that we did earlier. And then I went on to describe the amino acid transporter that was responsible for this phenomenon. And here it is. It's called B0AT1. It's the green entity. These are proteins. Each of these little balls is an atom. And these things bind to ACE2 and forms this entity. It forms a structure of 1, 2, 3, 4. It's a tetramer of these particular proteins that are embedded in the cell surface, in the cell membrane. And so here's a cell, and you can kind of see they're sort of studded along the surface of the cell, okay? Now, if you take a look at this protein in this region, what I've done is determine that these are stuck together, and we were able to determine that they form hydrogen bonds within about 3.5 angstroms apart. And there's a little sticky region down here, and it forms a bond about 2.5 angstroms apart. And I've also looked at the horseshoe bat genome, and it turns out that this region down here, this bond, instead of being a leucine to an arginine, there's a methionine. So it's very closely related in terms of how this happens in the intestine of other animals. But anyway, we determined that the structure of the ACE2, which is the receptor for the coronavirus, on these little purple regions on the top, that's where it's a sticky region where the ACE actually binds, is stabilized in the membrane by this amino acid transporter.
You lost me a little while ago. Can you tell me whether this is good or bad?
This is good. And I'll get a little bit later on here. Maybe this, see, this is a shill question okay? Because it's good, it's essential for life, because you need this amino acid transport protein to get nutrients from your intestine into your body. And the particular nutrient that I'm going to kind of hit again and again that's essential for you is tryptophan. Where's Trish? You know, what's tryptophan? It's an essential, it's an amino acid that is useful for a number of things. And we'll talk about how that's useful and how bacteria can alter that, especially in COVID situations. So this is, to answer your question directly, this is good, okay? And it absorbs these nutrients through this little channel, and the whole thing is stabilized with, by coincidence, the receptor for the coronavirus that sticks to these sticky regions on top. The spike protein from the virus sticks to the protein on the top of this receptor that's involved with nutrient transport, nutrient absorption, and specifically tryptophan. And I'll tell you why tryptophan is important in a few minutes. So let's talk about where all of this happens. So here's our gut, kind of lower part of your abdomen. There's the large intestine, the small intestine, and the intestine is basically a long hose. It's about 25 feet long or so. It's about maybe an inch or so in diameter. And if you take that hose and you cut it across section, that's what we're looking at down here, okay? So not looking down the center of the hose, here's our microorganism. This particular case, it's a coronavirus sticking in the center of the hose, the center of your intestine, okay? But the intestine is lined with cells. And here's one particular cell, and we're going to talk about what happens to that cell in a minute. It's like the lining of a hose. The plastic is made up of cells that lines the hose, and down the center of the hose go our nutrients and other things as well. And so if we take a look at this lining, and here's a cell, one cell, sticking out of the cell is our pink and green conglomerate that contains not only the ability to absorb tryptophan and other nutrients, but also has the sticky region that binds to the spike of the coronavirus, the SARS-CoV-2 virus. And it turns out that once this binds, that there's an enzyme that we're not going to go into too much right now, but it actually clips the spike, injects the spike into the cell membrane. That starts a series of injecting of the nucleic acid material from the virus, and the cell becomes infected. And that's bad. So this is the controversy. This is a good essential nutrient, and it has been hijacked by a bad guy. So the bad guy says, I'm going to glom on to an essential part that's required for life for the host. How evil can you get? Okay? And so this allows it to get into the cell. Now, back in 2020, Pfizer also reads the literature. And the wise scientists at Pfizer thought, you know what we're going to do? We're going to take the Stevens protein and co-express it with the ACE2 in an in vitro cell system and use that to screen our candidates for a vaccine for the virus. So here's a cell, and here's our good protein, okay, that Pfizer put into the cell. And well, I guess the PowerPoint kind of works here. So here, to blow this up, if we're looking at the top of the cell here, here's our amino acid transporter and our ACE2, which is, to us, is good, but it's being bound to the coronavirus through this spike protein. And what Pfizer did is they set up an in vitro screening system with thousands of cells, each one expressing this receptor, so that they could know. They would test various RNAs. They had hundreds of RNAs, and they try one and see if it prevented it from sticking. Try another one, see if it prevented it from sticking. And they finally hit the jackpot. And this particular one, the BNT162b RNA, was the one that ended up in the Pfizer vaccine, which through kind of Jungian serendipity, I happened to get in December of 2020, that came from the screening based on the fact that this amino acid transporter actually holds the receptor for the COVID virus. So, no, I didn't get COVID in 2020. I got the vaccine in 2020. So let's talk about, let's now kind of get into the concept here of how the gastrointestinal microbiome, even though COVID virus is part of our microbiome, there's an interplay with bacterial microbiome and viral microbiome in our gut. And so I'm going to return to this figure. Remember this? We showed it a minute ago. There's kind of your gastrointestinal tract down here and the cross section of the hose in your intestine. And looking down the center, it's made up of cells lining the hose. And it turns out that about a third of patients with COVID that do get COVID, maybe you've experienced this if you got COVID, about a third get gastrointestinal symptoms, and nobody knew why at first. Diarrhea, nausea, abdominal pain, vomiting, in addition to kind of the classic pulmonary functions of manifestations in the lung and fever. About a third of individuals get gastrointestinal manifestations, and nobody kind of knew why. I joined a team that looked at two populations of patients, patients with COVID and then healthy reference patients. And we looked at their blood bacteria and their gut bacteria through poop samples. And by poop sample, what that means is just a little bit like the size of a pencil eraser or a green pea, the end of your finger. It's just a small amount of poop. We can analyze the bacteria in that, and I'll show you how we do that. I'll show you how microbiome data comes about, in case you're wondering where that comes from. And we also looked in the blood. And here's some bacteria. I've just listed a couple different types of bacteria. And I want you to keep in mind something that Joan was asking me as we were coming in today, in terms of like, which bacteria should we favor and which ones are bad ones? That's not really the issue in microbiome. The microbiome is an ecology. It's a community of bacteria. There is no one single one that's responsible for anything in particular, either good or bad. It's the community of how those bacteria interact with each other. And so I just sort of listed a few of the many thousands of bacteria that we've identified here in the blood and in the gut. And it turns out, if you look at this pie chart here, that if you notice here, some of these have shifted. The pie distribution of the bacteria has shifted. And it turns out that these bacteria are leaking out of the gut in COVID patients and showing up in a bacteremia, in sepsis. So the inflammation in sepsis that is caused systemically or whole body wise, and including in the lung, comes from gut bacteria in a leaky gut situation that is part of the inflammation response in COVID. Okay, so what I want to do now is talk a little bit about how we identify microbiome. The general public just sees, you know, that you look on your yogurt thing and there's some bacteria in there. Well, how do people know what they are? How do you, if you're part of one of my studies and you have a poop sample and we analyze it, how do we determine what that microbiome is? And I want to explain that a little bit. I'll tell you what we don't do is we don't culture it on Petri dishes, okay? We don't identify bacteria individually. Instead, we do this genetically by looking at the bacterial nucleic acids. And as you know, the nucleic acids, whether it's bacteria or people or any living thing, is made up of a double helix of nucleotides A, T, C, and G, adenine, thymine, guanine, and cytosine, okay? And these form this ladder. Just to remind you here, these are the actual chemicals. So I'm doing this for some of the scientists in here that recognize nucleic acid chemistry, that there's A, looks like this, adenine, thymine, guanine, and cytosine look like this. And we analyze this from a poop sample and get millions of these nucleic acids, A, T, C's, and G's, and the data looks like this. So we get, and so if we have one bacteria species and it's got a gene, and here's its nucleic acids, and here's another bacteria and its nucleic acids. Okay, quick, what's the difference between those two genes? Can you tell me if there's a, well, I can tell you there's a single nucleotide difference here. And the PowerPoint's not going to show it, but it's up. There's a G here and an A here that's different. Otherwise, they're identical. But from a genetics point of view, those are two different genes. They can express two different proteins in a different way. So we have to identify all the nucleic acids in all the bacteria of all the patients. And what we do here is we're looking at specific genes in an individual. So let's say there's a patient and you were in one of my studies here. And we have a big matrix of data, okay? And some of this matrix of data is three-dimensional matrix, includes specific genes from the bacteria that I've shown you. Well, let's put that in the points. And then each gene has its own unique nucleotide sequence. So right now, you can imagine the matrix is starting to get kind of complicated. because from each gene and each nucleotide source, that comes from a different species of bacteria. So let's identify the change in the bacteria by different colors. The different shapes might represent the nucleotide sequence. The position on the matrix might be the specific gene. Then there's metabolism, the bacterial metabolism, whether there are neurotransmitters that affect our mood or there's chemicals that affect our blood pressure. We look at that. And so we say, we take each point and the degree of tilt is the metadata that has to do with our physiology. And then not only that, but there's a relative abundance difference in these bacteria. So the relative size comes into the matrix. So there's about an 8-dimensional matrix here. I don't know about you, but my brain can't think an 8-dimensional matrix. And not only that, but this is one patient. Let's say we had 2 patients and we wanted to compare them. How are you going to do that? Not only that, but if we have 100 patients enrolled in our study, how are we going to sort out this gajillion dimension matrix? And the data starts looking like this after a while. And Alan, do you remember the days when your data started looking like this? It was just a conglomeration of how are you going to sort out this raw data? Well, what we've done is develop artificial intelligence machine learning programs that will sort out this data, put it into the hopper here. It comes out around and around it goes. And now my brain can wrap itself around the data. So instead of 10 gajillion data points from the nucleic acids, A, T, C, and G, the relative abundance, that sort of thing, it has sorted it out. And I'm a simple-minded guy. I just want to think blue, green, red. That's all I want to know about what's going on with my data. And lo and behold, that huge matrix will tell us that there's a cluster of people in our study that have hypertension, and they all have a common bacterial signature. All their ATCG nucleic acids have a common signature. Another group with hypertension. And then there's another healthy reference group. And each of those are cluster, and we can identify individuals based on their bacterial communities, not individual microbiome, but the communities of the hundreds of bacteria. And so what we do now is we know that, let's say, this particular study with all those bacteria, all those nucleic acid sequences, cluster individuals and give us a nice, tidy cohort that we can work with. And so we take the cohort. In this particular study, this one showed that there were individuals that were clustered that have depression. Another group of people in the study, their bacteria clustered them, showing that they had hypertension. There was another group, interestingly enough, that had hypertension, high blood pressure, and depression, and they had a completely different microbiome set than the other groups do. And then our healthy references were completely separate from everyone else. Now, how are you going to know what microbiome, what bacteria are in each one of these and what they're responsible for? What we do is we put these together initially in something called a heat map. If you look along, I guess the resolution is okay here. This is a microbiome community. These are bacterial species that come out of the ATCG nucleic acids. We can identify what the different species are and the relative abundances of each of those microbiome species, and then map those according to color intensity. So for example, these particular bacteria up here are shown in high density, green, whereas these bacteria over in this cluster with this bottom row, red, are a low density, okay? So you're going to do a little experiment here. You're going to tell me which bacteria, which microbiome is responsible for hypertension as compared to healthy reference individuals. Now this is where, see, I'm the simple-minded guy. Colors, green and red, okay? I don't care about the 10 gajillion matrix, we're just going to look at green and red and the relative intensities. So now there's here, but we're going to focus on, just to make it easy, hypertension and our healthy reference. By the way, I don't like to call these people normal. Okay, I teach medical students, nobody's normal. Okay, everything is a reference.
5-minute warning for the second service.
Get up and go, that's fine. So if we look at the relative distribution, the green and red distribution, we can see, for example, in the healthy patients, they are lacking this microbiome population. And it just turns out these are bad guy bacteria. But they have a high density of these microbiome bacteria, which turns out to be good guy commensal bacteria that we have kept with us for 40,000 years. Now let's look at patients with hypertension. The same group that we've analyzed their ATCG nucleic acids, this group right here. It turns out that the bad guy bacteria that are low in our healthy population are quite prevalent in the hypertension group. And not only that, but the good guy bacteria in the healthy reference group are low. So the chemical warfare between the good guy bacteria and the bad guy bacteria is slanted toward an unhealthy state in the people with hypertension, as opposed to the reference healthy individuals that have good guy bacteria that kind of dominate the microbiome community in their body as meta-organisms. You want to give me an advance here? Okay, so what we do, we put it all together and we can identify the 4 cohort population here. And I've published that this is an ecosystem. Remember, it's not an individual bacterium in your microbiome that's responsible for you being a healthy individual or having some kind of an unhealthy state. But it turns out that this is the dominant community in your body, of your meta-organism body, so that healthy individuals happen to have a higher prevalence of these bugs, and individuals with depression happen to have these particular bacteria as their microbiome. Individuals with high blood pressure, we note, have these bacteria. And it turns out that individuals with depression and high blood pressure have a completely different microbiome profile. Their community and their meta-organism structure of your body is completely different. And we published that I think we may have identified a new disease called depressive hypertension. I can tell with 95% accuracy based on your poop sample whether you have a depressive condition, anxiety, hypertension, or depressive hypertension, or whether you're in a healthy state, based on the ATCG nucleic acid sequences in that huge matrix and the particular bacteria that are sorted out from all of that. Yes?
The DASH diet that has lots of fruits and vegetables in it that decreases people's hypertension.
Glad you asked. The question here is, what about dietary interest? And does it affect this? And the answer is yes. Details in terms of tryptophan. What is tryptophan? It's an amino acid. I'm putting a nutritionist on the spot here. Okay. So we'll be getting into that. Okay. So let's now go back to this concept of microbial communities as part of our microbiome in our gut. Remember this picture here? This is our gastrointestinal system. We take a cross section of the hose and we're looking at what's going on in the center of our gut, and especially on the surface of the hose. Like, if you cut the hose, in half, it will look like this. If you cut the hose the long way, then what we're looking at is the lining of these cells. And so I want to do that now, is look at the long way. So if this is the hose cut the long way, that here's our layer of cells in the intestine, okay? They're all, and the cells are connected together by what are called tight junction proteins. And in the lumen, so remember that looking down the center of the hose, if we do a longitudinal longitudinal section, this will be the center of the hose, the center of your intestine, okay? And within that are various nutrients, Twinkies, whatever your nutritionist recommends that you eat, that it will be in the lumen of your gut, in addition to the particular bacteria, whether it's good guy bacteria or bad guy bacteria that are stuck to the wall, primarily very close to the surface of the wall, the lining of the hose, like the green plastic part of the hose. The bacteria are mostly stuck to that rather than floating around in the center, but I'm showing you kind of near the surface here. These are bacteria, the gut microbiome, and they either the bacteria produce tryptophan and all the other 20 amino acids, but I want to focus, again, I'm a simple-minded guy. We're just going to stay with one amino acid, and we're going to talk about tryptophan. So what tryptophan, remember the picture I showed you early on when Allison asked me, is it good or bad? Well, it's good because tryptophan is absorbed into our body by this transporter that we characterize in my lab, and is stabilized in the membrane by ACE2. And in this case, there's no COVID around, the ACE2 amino acid transporter complex is good for you. Okay, Allison? It's good for you here in this case. And the reason it's good for you is it activates some mechanisms in your body that causes antimicrobial products to be made. So this is part of the coevolution. These bacteria are smart. They've known enough to reside in a host that allows them to create chemical warfare against their bacterial foes and will ward off the bad guy bacteria. Okay, this is part of the ecosystem of our microbiome. Not only is this involved with the microbiome to maintain a healthy internal environment, but the tryptophan then goes into the blood because our intestines are enveloped by blood vessels and goes on to the rest of our body. And tryptophan is the main precursor for serotonin. Okay, the gut is the primary source of serotonin in our body. About 80% of the serotonin in your body comes from your gastrointestinal tract as modified by gut bacteria. Okay, not only is that, but also the tryptophan will go to your liver and it makes niacin to prevent certain diseases that require niacin. And tryptophan also activates a hormone in the intestine called GLP-1, which goes to your pancreas to release insulin. So the good guy bacteria modulate not only neurotransmitters in your body, but the good guy bacteria modulates your blood sugar as well through co-opting its host to keep it in a healthy state, because these good guy bacteria want a healthy home. They keep it that way by keeping the host healthy, by maintaining a healthy blood sugar level, by maintaining a healthy mood state, for example, and serotonin does some other things as well. Now, let's say Darth Vader comes into the picture here, our coronavirus, okay? And coronavirus now will bind to where? It binds what was a good player, now becomes co-opted and hijacked as unwittingly a bad player in a disease state. This is pretty evil that this is co-opted a necessity protein in our body. And it binds, and what happens is that by binding this complex, we've shown that it interrupts the interaction and the properties of amino acid transport. So our tryptophan molecule then is not absorbed as well. Where the healthy state, it was absorbed, it's not. If tryptophan is not absorbed very well, then that cuts off the supply of antimicrobial products, and you'll get an overgrowth of bad guy bacteria, which happen to disrupt these junctions between the cells, causing leakiness. And I'll show you what that leaky gut problem is by disrupting the wall of the intestine. But not only that, it interrupts the production of this hormone that would have regulated insulin. So one of the problems with COVID, as you may know, is people with type 2 diabetes are especially susceptible because of the disruption of glucose regulation through GLP-1. So what is one of the mainstays of COVID treatment for individuals with type 2 diabetes? To give mimetics of GLP-1 like Trulicity or Ozempic. These drugs stimulate insulin in the same way that GLP with these semaglutides will mimic the cutoff that the bad guy bacteria have caused because of the COVID infection. Now, the problem with having a leaky gut is that not only do we have leakiness of materials like bad guy chemicals that the bad guy bacteria make, but the bacteria themselves get into our blood. And this was remember the pie chart we had a minute ago, that the shift in blood bacteria goes toward gut bacteria that causes an overall inflammation from the microbiome in our gut that had been kind of triggered by Darth Vader here. Now let me take that same slide and I'll put it over here. And so if we're looking at the disruption of gut metabolism, and the insertion of bad guy bacteria in our blood, that leads to, well, I guess the PowerPoint isn't bad here. That word says inflammation. It leads to a whole body inflammation. The response of circulating bacteria that came from your leaky gut as caused by the disruption in the junctions between the cells. And this not only causes a whole body issue, but also causes inflammation not only in the lungs, but everywhere in the body, and will lead to GI symptoms of COVID, the nausea, vomiting, and diarrhea. And it turns out that because this is a very high prevalence in the gastrointestinal tract, that the coronavirus shows up in the stool. And so with a group at UAB, we published a paper back in 2020, kind of alerting people, you know, this is a big deal in the GI tract. I think you could have spread of COVID possibly through the sewage system. Well, it turns out that the virus doesn't really survive to be infective, but nevertheless, as you know, the main surveillance mechanism for COVID is to look at the virus nucleic acids in the sewage effluents and in water treatment plants. So this is done now routinely instead of looking at the hospitalizations. Now COVID surveillance is done by looking at water treatment plants and how much of the virus has been shed and shows up in the sewage system because of shedding the virus from the gastrointestinal tract. Now let's talk a little bit about the gut microbiome in blood pressures. We know here from that last slide that the leaky gut causes bad things to go into your blood. And we've measured a number of things. I put this in here for Alan and some of the other scientists in here. So you know this is real data. It's not just Fox News comments or something. It's real data. That there are a number of markers of leaky gut. And I'm showing here that they go up in our COVID cohort. And if you graph blood pressure, this is systolic blood pressure, as a function of these leaky gut molecules, there's a direct correlation, a positive correlation with the leakiness of the gut. And also, by the way, these same molecules are involved with the blood-brain barrier leakiness as well. So not only do the molecules get into the blood and cause inflammation, but that they can cross blood-brain barrier and cause some problems in the brain, which we'll talk about in a minute. And in terms of the brain regulating, the brain regulates blood pressure. There's regions in the brain that regulate, as you know, cognitive events and mood. The brain also works with the kidney, and and the lung and some other organs in the heart and the vasculature to regulate blood pressure. So if the inflammation occurs in regions of the brain that are regulating blood pressure, it will disrupt that. And there's a direct correlation between how leaky the gut is from its microbiome of bad guy bacteria and blood pressure in individuals. So let's kind of start winding this down and talk in terms of like we're putting some of this together, some of the topics that we've covered today here. And to begin with, I hope you're remembering here that we're meta-organisms. We're a conglomerate of crosstalk, intertwined people cells and bacterial cells. And we've come to this point in time because of the evolutionary pressure favoring this system, that the bugs keep us out of trouble and we repay them by becoming a favorable housing situation that the bugs can then go on, the next generation can live. And part of the collateral damage of all this, and I've given one example, it's hypertension, is that there's we're still on the path of evolution. You see my arrow is continuing on. We're in the middle of evolution. Maybe some point in time, the collateral damage problem will have been solved, but in the meanwhile, we have a relationship with our microbiome. So this is going to be difficult to show because I think it was kind of corrupted a little bit in the PowerPoint, but this early slide, I think it was the second slide that I showed here to kind of wrap things up, that we have our gastrointestinal tract, we have our brain, and I'll show you in a minute. Down here was our whole body physiology, kind of the rest of us, and that there's a two-way communication between these. Yeah, it's not going to work. There's a two-way communication, and part of the events that occur in the brain occur because we have centers that are involved with mood regulation, and there's centers in the brain that are involved in blood. Can you see that word underneath there? It says blood pressure regulation. Our brains control blood pressure. And the gut, well, Elisa showed the neuron. The gut has a nervous system itself. It's called the enteric nervous system. Some people call it the little brain of the body. The gastrointestinal tract kind of is a standalone nervous system that can think on its own. Sometimes it does some things that you don't like it to do. And your gut has its own standalone nervous system that communicates. Well, there it is. There's the two-way arrow. It communicates with the brain. And it does this communication through neural connections and hormonal and chemical connections. The neural connections would be through the vagus nerve, which is a parasympathetic nerve, through various sympathetic nervous systems, and then there's some nerves in our lower spine area that are like the splanchnic nerves that regulate. There's a communication between the gut and the brain through these nervous connections. But not only is there the neural connection, but there's also small molecules that are exchanged coming from the gut that react with the brain. And some of those are neurotransmitters, like our tryptophan metabolite that we talked about a minute ago, serotonin. There's oodles of other molecules, dopamine, GABA. There's now this gets into Trisha's question here in terms of fatty acids, and our diet can control the brain events coming from the gut, and the gut bacteria modulate these particular molecules that tell the brain what to do. And not only the brain, but the whole system physiology. For example, red meat contains a nitrogen compound that your gut bacteria convert into trimethylamine, and then that goes to your liver, and your liver creates a chemical called trimethylamine oxide, TMAO. TMAO is the part of red meat that causes hardening of the arteries. It comes from bad guy bacteria converting those components into artery hardening molecules. Okay, so if you can control your diet, you can lower, for example, TMAO. What about fatty acids? There are certain very useful fatty acids, so-called short-chain volatile fatty acids, like acetate, butyrate, are the main ones. And so certain foods like prebiotics that contain a lot of fiber are loving it by the good guy bacteria, and they produce certain fatty acids like butyric acid, butyrate, which help tighten the tight junctions in this wall of the gastrointestinal tract. So those small molecules play a role in whole body physiology coming from the gut microbiome modifying our diet. The bacteria shed into our system from the leaky gut are a source of antigens which turn on inflammation. We don't have time to show the mechanism here, but that amino acid transporter and angiotensin converting enzyme in the gut regulates sodium absorption. And so the regulation of sodium in turn affects blood pressure by changing blood volume, and various other nutrients, whether it's Trisha's Twinkies or not, can affect the ecology of good guy versus bad guy bacteria and their metabolites, which then go on and affect the rest of our body. Question over here. Yeah. Now I think that you're supposed to ask the questions at the end, but if this is appropriate, then go ahead and interject it now. I think this is good.
Talking a lot about leaky gut, and I'm wondering, does everybody have a leaky gut?
No.
And are we born with a leaky
No, no, the leaky gut is a pathological situation. She wants to know whether, we'll get into kind of a general question and answer thing in a second. But just to kind of that particular topic, your gut normally has a tight junctions. It's the normal healthy reference state is to have a non-leaky gut. There's a division between your internal part of your intestine and your blood. And it's what moves from your gut into your blood is regulated by the amino acid transporter, and there's lots of other transporters too. There's dozens of them that regulate. They're like gatekeepers that regulate it. But if the walls become wet tissue paper, then all the regulation goes out the window, and it's just everything just kind of leaks on its own. Okay, so so what we're talking about here is the axis between the gut, its bacteria, its microbiome, the brain and its various centers in what it does, and how the interaction of these things regulate whole body physiology, whether it's our lungs, our immune system through interacting with spleen and bone marrow, regulating electrolyte and fluid by controlling how our kidneys work. Okay, and all of this comes from this triangulation. See, I have arrows with two heads on it. There's a two-way street. They each control each other. Our brain, Joan was asking me this on the way over here. Does our brain control what's going on in our gut, or is it always from the gut controlling the brain? There's a top-down control too, because for example, your brain controls, and your central nervous system and your other hormones control the blood flow in your gut. So if bacteria can control your blood flow to your gut, it can control whether there's oxygenation in your gut and regulate whether you have a prevalence of what are called anaerobic bacteria versus aerobic or facultative bacteria. So bad guy bacteria, which may be more anaerobic, will die off because the blood flow may have been turned down, so the oxygenation state in the gut wall is lower. How did that get turned off? Our good guy microbiome have regulated our system, and perhaps to the detriment of our blood pressure, that it may cause vasoconstriction in the gastrointestinal tract. The benefit is it may kill off the bad guy bacteria. The collateral damage is we may exhibit whole body higher blood pressure because of that. So let me just summarize what we've talked about here today. We kind of introduced this concept that people are meta-organisms. We're a conglomerate of intertwined human cells and bacterial cells. We talked a little bit about one microbiome niche, that is the coronavirus and its relationship to gut microbiome. And then we kind of got into the fact that the microbiome and the community of bacteria steers our whole body physiology. And I gave you a couple little examples of how it affects blood pressure. It can affect mood centers in our brain, and just generally overall health and inflammation state. So let me just leave you with this last slide. Since we're at UCG and this is sort of a philosophical group here, I want to leave you with some thoughts about our gut microbiome and people being meta-organisms. Okay, these are some thoughts you might want to think about in terms of what we've been talking about here. So if you eliminated half the cells in your body by, let's say, a antimicrobial cocktail where you completely nuked half the cells of your body, or X radiation and plus chemotherapy, so that you eliminated half the cells which are compulsory for human existence. And by the way, this is done in animals. You can raise what are called notobiotic rats and mice and lab animals. If you raise them, they won't live past the neonatal state. They'll die. They won't survive. If you take those same animals that have no microbiome and you spike them with a little bit of good guy bacteria, they go on to be healthy adults. Bacteria are absolutely necessary, as proven by your existence today in the coevolution of, we have bacteria that have co-evolved with us as meta-organisms. So the one question is, if you got rid of all your bacteria, would you cease to be a living entity? And maybe more to the point, what does this mean to be a human being exactly? If you're a meta-organism, what does it mean if half of your cells are prokaryotic bacterial cells? I mean, what is the concept of being a human being? And maybe even more to the point, who are you anyway? So anyway, now I'll entertain some questions. You have a mic. Yeah, I wanted to see who's there.
Relating SARS-CoV-2 to the whole system, of course, the main influence that we see on the outside anyway, people suffer mostly from lung problems.
Right.
Not gut problems.
Right. About a third of the people have gut problems, but almost everybody else will also have lung problems.
Right. But yeah, so it seems that the fatal effect is often the binding to the ACE2 in your
Well, that's a little bit of a misconception, that the effect is binding to the ACE2 in the lung, in the pneumocytes. There's pulmonary epithelial cells that have a similar situation. But it's also the inflammation that comes from what's called a cytokine storm that arises because of bacteremia, the sepsis, that is of gut microbial origin.
Okay, that makes lots of sense.
Okay, not only that, but I've only given you kind of a little piece of the story here, and that is the lung also exhibits an ACE2 stabilizing amino acid receptor that transports proline and glycine, which our lab discovered as well. That is in the lung and in the intestine and in the pancreas, and it's in a number of other places in the body as well. So the story is kind of complex. Physiology is kind of complex, and you can't just look for the keys underneath the lamppost. You have to kind of have a broader kind of perspective to.
One other little question, and that is, when you receive an inoculation with the vaccine, the mRNA vaccine, then the effect, I know firsthand, is often on the gut. You have a transient gut problem.
You can. Some people do, yes. Part of that problem has to do with the fact that the gut has its own enteric nervous system. There's a gastrointestinal nervous system, and the inflammation response to the vaccine kind of excites that. In fact, when I got vaccinated, the first thing I did is ate potato chips and jelly donuts and soda because I wanted to get my inflammation state up. I wanted to have the worst possible pro-inflammatory body condition possible. I didn't tell Trish because I know she would scold me if I did this. So it was Twinkie City when I got my vaccination because I wanted to make sure I was in a highly inflammatory state to get the best possible response to the vaccine.
And another effect is on the sodium flux.
The sodium is very complicated because the sodium involves what's called the renin-angiotensin system, which involves the lung and the kidney and the brain. There's a very complex system of regulation of sodium ion. It turns out that in the intestine, that's the main source of your sodium absorption, and there's a transporter. There's several transporters that transport glucose and various amino acids with sodium, and when those are disrupted by the peptides that are formed by the combination of ACE, angiotensin converting enzyme, and ACE2, the homologue of that, that disrupts sodium and electrolyte balance. And by the way, just so if any of you are on like ACE inhibitors for hypertension, angiotensin converting enzyme, that's not the same as ACE2. ACE is a kind of, it's kind of a detrimental protein enzyme. And ACE2, it's a good guy enzyme. It's a good guy enzyme that takes the products of ACE and breaks it down to prevent the ACE. And there's research now going on with hypertension research that focuses on revving up ACE2. But just to kind of keep in mind, at one point there was some in early 2020, people were wondering, should people with hypertension continue on their ARBs and their ACE inhibitors if they have hypertension? It turns out it actually would be beneficial to stay on those because you want to keep the renin-angiotensin system kind of at bay to prevent disruption of sodium imbalance.
I have a question. Bruce.
Yeah.
This has been a wonderful talk. You know, as the author of our COVID updates, this really crystallizes. I want to reflect on a study that was done about 30 or 40 years ago showing how infants, human infants, when they're first born, acquire the biomes of intestinal biomes of their parents when they return home. Now, how important is that?
This is very interesting. This is an active area of research. Actually, at UF, there's a little bit going on with Joe New in neonatology, that we acquire our gut microbiome from our communities. And the transmission of those bacteria and those states of health or disease are a familial trait. Okay, familial, most people think is a genetically translocated trait, but familial means it stays within your family. And a lot of disease conditions are because families are cultural and they eat the same kind of foods. They're kind of all kissy face and exchange the same bacteria that stays in that kind of tribe. And that's propagated generation to generation. And when those people move, their health changes because they've kind of stopped eating that food, whether it's fatty food that promotes bad guy bacteria and everything. So where do these bacteria come from? Your question is, where do they come from in the first place? They come from your mother, okay? And kids that are born with C-section now, in some settings, will actually take vaginal swabs and smear it all over those babies to make sure that they get inoculated and they get their first dose of their microbiome that's going to stay with them for the rest of their life. I have found that in individuals that take antibiotics or have a skewed microbiome, you come back to the center. You kind of have your microbiome, it's sort of yours, for better or worse. And you can skew it. By the way, some ACE inhibitors that control hypertension actually have antibiotic properties. They kill off some of the bad guy bacteria that are causing hypertension. This is a new revelation. They not only affect the renin-angiotensin system, but some of the ACE inhibitors actually have antibiotic properties. And some some antidepressants have antibiotic properties as well. In fact, that's what got me into this whole thing in the beginning, because there was a study at in New York of a state mental hospital where individuals with schizophrenia were going to the infirmary and getting treated for like eye infection and some other infections with a drug called minocycline. And their psychosis cleared up and nobody kind of knew what happened. And I thought, wait a minute here, you know, maybe what it's doing is it's altering their bad guy bacterial profile. And it's turning out that that's true, that a lot of mental states and have to do with neurotransmitters that are altered by your gut microbiome. And so now there's research going into probiotics that actually can skew your ecology, your kind of organism ecology in a favorable direction. And let me just interject one point here, that a lot of people have the misconception that if they take a probiotic, it's a silver bullet, okay? You know, Culturelle or VSL3 or whatever your choice of probiotic. There's very few probiotics that have any of the research that they haven't pulled it from our papers, I can tell you that. They just kind of make up some bacteria, put it in, tell you it's good for you without any foundation for it at all. And I've also, in the patients that we studied looking at their gut bacteria, I know for sure certain people in that study, my family members and myself included, that were patients in the study, take probiotics. None of those bacteria showed up in the poop samples. Okay, so what happens is the probiotics are players in the community of bacteria. They aren't necessarily the silver bullet that's going to cure you. They're players in a community of a microbiome. And getting back to your original question, to set the stage for having your microbiome be what it is, it comes from your mother. Okay, or it comes from your environment early on in your health. There is a little bit of research that show that there's possibly bacteria that crosses the placenta, and I know one study that showed that there's at least gram-negative bacteria that show up in the brains of fetal tissue, and it's thought that perhaps it crosses the placenta and can get into the developing fetus. And so you may have a start, but it's not a lot. It's just like, if you look at the slides, there's just little speckles here and there. It's not like, you know, like 100 trillion cells or something. It's just a little bit. But you get your microbiome from your mother as you're born, primarily, whether it's from breastfeeding or whether it's from the birth canal or just the environment that you're in, you get it from your mother. I think Rolf had a question, and we'll go back to Allison over. Say that again? Oh, okay. Why don't we get Rolf, and then we'll get the big board.
Can you have a biome without viruses?
This is a very interesting concept. Can you have the biome, the microbiome includes all microorganisms, including yeast cells, which are eukaryotic, bacteria, which are prokaryotic, and viruses. So the microbiome includes viruses. There's a whole world of bacterial warfare that occur with what are called bacteriophages. Okay, bacteria have an interaction with the community of their bacterial hosts. And there's a lot of research now going into trying to identify specific bacteria that can kill specific viruses that are silver bullets that can kill specific bad guy bacteria that can help your whole body physiology. So viruses, to answer your question directly, viruses are part of your microbiome. By the way, microbiome means the genetic component of the microbes. The microbiota are the actual bacterial species and viruses. Okay, so there's a difference between, I'm primarily focus on the microbiome because we identify those by nucleic acids, A, T, C, G nucleotides. But it's the microbiota that are responsible for their microbiome interacting with your physiology. Why don't we take this the board here, and then we'll get back to some audience questions. Dean Chance, George. I talked to George about this at the men's Christmas thing, I think. But anyway, you wanna pull up one of these and have them ask a question? Do I read this? Okay, I'll read this with a complex. Okay, why don't we? I'll quickly go through these and then I'll return to field some questions here. Okay, I'll go to the chat to the chat string here. So John is asking, with the complexity, what are the benefits of taking a single lactobacillus? Well, I think I kind of answered that question, that there's no magic bullet. If you looked on the ones that we've identified, that lactobacillus actually as a genus is actually prevalent in healthy individuals. So it can help push your microbiome in a healthy direction, but it's not going to be a silver bullet. As a general practitioner, can order a microbiome test. I would say it might be interesting to order a microbiome test. There's not a lot you can do with it at this point because it's really, we don't have the tools to alter the microbiome, the community of bacteria. And so it might be interesting, but there's not a lot of practical application of what you can do with that information once you have it. I just did this. I talked about bacteriophages as part of the microbiome in terms of adjusting your microbial community. So George, I hope we covered your question. I talked about phages. What is the natural bioidentity hormones? Oh, that's a very interesting question. Bacteria take your food and break it down into peptides that are bioidentical. In fact, one of the reasons why milk is antihypertensive is because the casein protein in milk actually is broken down. And I have a slide on this, but I'm not going to go to it. Breaks down the casein into a peptide fragment that mimics the product of ACE2 hydrolysis in the intestine, which lowers blood pressure and reduces sodium absorption. So there's some other effects of like cholecystokinin, which is a hormone of the gut, that bacteria make from the food that you eat, from the protein you eat. So biosimilar molecules come from the microbiome of your gut. What are we here? Is there one way to transform from an unhealthy to a healthy state? Mediterranean diet will go a long way in keeping your good guy bacteria happy for the many reasons we talked about. Fecal transplants. So fecal transplants are useful for kind of one thing primarily. Fecal transplants take a healthy gut microbiome and you do a poop transplant into an individual. It's primarily one condition, and that is C. diff, okay? Clostridioides difficile is a very difficult to treat with antibiotics. So if you can harness the community aspect of good guy bacteria and dominate your gut in somebody with the C. diff overgrowth, they can kind of kill off the C. diff. And so fecal transplants are useful for that. There was a phase where there was an infection in one of at Penn State where they did a fecal transplant and the patient died. And it turned out that they traced it to the donor who had a disease that was I think it might have been hepatitis C or something that actually went into the sample and the patient. So they put a clamp on the fecal transplant. It's back online now, and fecal transplants are being done. Let me now. There were some questions here, Alice.
That antibiotics are targeted, otherwise you kill half your body.
Antibiotics are like hitting a nail with a pin with a sledgehammer, because of the collateral damage of getting rid of good guy bacteria that kind of go out with the bathwater with the bad guy bacteria. So antibiotics are a little dangerous in the sense that you've kind of jumbled up your microbiome ecology. And it takes a while, weeks, to kind of come back to the center again. And you can kind of promote that by taking a probiotic during your antibiotic therapy, like a lactobacillus, like Culturelle or something. But it's not a silver bullet. But I think what you're asking is, what is the effect of antibiotics on overall health? And some of them can have detrimental effects because they skew the microbial population.
On the other hand, antibiotics make people live longer, right? They're not dying of.
Well, antibiotics, yeah, antibiotics. I mean, if you have a whole body sepsis and an individual dies because of whole body, your other organs are affected, like say you have liver damage or kidney damage, brain infection. Yeah, antibiotics, it's like everything in medicine, and Alan can tell you this. You have to cost benefit weigh, whether it's going to be beneficial to you or not.
I'd like to thank you for making science visually stunning and humorous.
Okay, well.
And comment, that it hurts my heart a little bit to hear about organisms being or bad guys or villains. And so, I just bring that up.
Are you a microbiologist?
I have teaching geriatric medicine fellows in last week's Journal Club. We reviewed the Journal of Neurology. They had a very poopy article that showed the microbiomes from Britain and a link between laxative use and all-cause dementia. And so I wanted to ask your opinion. I don't need that. The laxative use is going to increase motility. It's going to cause kind of a flush out of your bacteria. And if your microbial community happens to kind of go out the back door with the laxative use, that is not a good, healthy situation, because it may be dominated by what are called film-forming anaerobic bacteria. Bacteria aren't floating around the center of the hose. They're clinging to the 50 micron mucus layer of the back of the gut. And so if you flush out non-biofilm forming bacteria, which are good guy, then you're going to get dominated by the biofilm ones, which generally can be actually resistant to antibiotics, as you may know. The biofilm forming bacteria can be harmful. And I'm glad you brought up this point about good guy and bad guy, because that gets into a binary mentality about viewing the world. And I've tried to emphasize the fact that the microbiome is a community. It's actually kind of an interactive community. And I don't know how you would characterize the SARS-CoV-2 viruses, whether you think that's a good thing or not, but I would say that that's kind of on the kind of Darth Vader side of microorganisms. It's not a good thing. But in terms of labeling, I guess you'd say it's a pejorative label to label a good or bad, but it certainly is not. It's certainly a harmful microorganism, the SARS-CoV-2 virus. Does that address kind of what you want to know? Yes.
Well, a lot of information, wow. And it puts it in some kind of larger perspective I've never thought of.
Who are you anyway? What happened? Am I bacteria or am I people? Go ahead.
I have a practical question. Obviously, you do an anti-inflammatory diet, right? Mediterranean dash, something like that. I'm also thinking about body position. Like the gut is here, the brain is here, we're standing here, but animals were like this, and they were like horizontal. So they were more maybe balanced or different. And then my question is, I'm teaching a form of yoga that is a lot of inversion. There's a lot of going down in different ways with different postures, and that's the main focus. And so I'm wondering what you think about body position physically as to the physiology of
It's important because your gastrointestinal tract, even though it has its own enteric nervous system that controls motility, and there are hormones that control motility, and your brain controls motility, and the gut bacteria control motility, there's still, it's smooth muscle, and smooth muscle under the control of the sympathetic nervous system has limitations. And so if you have a physical stasis, then that's not going to allow communication from one chamber of the gut to another chamber. So I think posture, movement, exercise are all, they kind of go hand in glove with a Mediterranean diet in terms of keeping a healthy gut.
But actually, the upside down position can be very restful.
Well, it may be restful for a lot of reasons that don't have to do with your GI tract, other than maybe relieving pressure that at an unconscious level you didn't know was kind of bothering you. So for that reason, it has an effect. And then there's this kind of secondary kind of gravity effect to keep motility, keep things moving around. There's a sort of secondary effect rather than the psychological effect of kind of pressure relief, let's say.
There's a difference in receptivity for receptors, actually, right?
Right. Your gut is just laden with receptors. There's an interesting property, but you can slice the intestine in half and you won't feel it. Okay, there's no feelings, but if you stretch it, then you'll feel it. You know what that feels like if you have a stretch, whether it's distension from bloating of some kind or overeating or something. Stretching, there are stretch receptors. There's millions of stretch receptors that could be redistributed by posture, which you're promoting in your yoga positionings.
Yeah, and this method is all about inversion, so it's very interesting.
It could certainly affect your mental state by getting your gut community in a healthy interaction. Definitely.
So the back bending and the twisting and all this.
Yes.
Yes, the small intestinal bacterial overgrowth test and resulting information you get from that, is that helpful?
Okay, so the gut is made up. It's a continuity system. Starts in basically your esophagus, and then your stomach, and then it goes to your small intestine, and then your large intestine. And the large intestine is generally kind of isolated from the small intestine. There's a valve between the small intestine and the large intestine called the ileocecal valve, and that prevents backflow. And the wiring of your nervous system in your gut is such that it's a one-way movement. Things kind of go on a conveyor belt from top to bottom. Okay, it's called oral to aboral end. And generally, in a healthy situation, your gut is a continuously moving food and content from the top through this valve into the large intestine. But occasionally, there's a problem with the valve, and you get a retroactive reflux from the large intestine, the right side of your large intestine, into the small intestine. They're two different organs, but those bacteria don't belong there. They go up, and they can cause an irritation of the immune system, which is in the wall of the small intestine. There's a whole immune system there, and cause inflammation. One of the problems with that, if you have bacterial retrograde overgrowth, that's different from leaky gut. Okay, so your question kind of has two parts. There's retrograde flux of bacteria causing what's called bacterial overgrowth in the distal small intestine. That's one aspect of your question. And the second one is the wall itself. Is it leaky, allowing bacteria and bacterial metabolites to go into your blood? There's two parts to your question there. And I kind of addressed the first one, and that is just the location of the bacteria themselves back into the intestine, causing inflammation. Then there's a second aspect of that, and that is the leakiness from the inflammation going into your blood. Does that address anything that you wanted to know about? Sorry, the what?
The SIBO test, because I did the SIBO test and it was kind of extra. And then they give you a diet. Does that remedy it?
It can help. Part of the diet increases your motility to kind of, it's called the intestinal housekeeper. Every like 2 hours, your gut has a single sweep, which keeps things moving south. And your diet, if especially has a little bit of fat in there and some tryptophan, which activates a hormone called cholecystokinin that increases that motility to kind of promote that one-way direction. So your diet can help in that way. Plus, the diet will increase if it's a diet that has certain kinds of fiber and it's a Mediterranean diet with good proper oils with the right kind of hydrocarbons on the oils. It'll produce short chain fatty acids like butyric acid, acetic acid, propionic acid, which tighten the tight junctions in the wall of the intestine, like the plastic part of the hose of a hose. It will keep those tight junctions, the fatty acids from the diet that the bacteria make. Does that kind of address what you wanted to know? And we'll wrap it up. Okay, you can ask me. Yeah.
Just a quick one. First of all, this is not a very scientific. I am so amazed. I have not ever heard such a wonderful presentation.
Did you learn something?
I mean, I'm come from the sort of English or other language, but I was, with the beginning, you're talking about the HR precursors way back thousands of years.
Yeah.
So all the bacteria continue on to now, right?
This is very interesting because if you look at the evolutionary biology and the evolutionary genetics of certain species of bacteria, they emerge in time coincidental with the emergence of primates and of mammals in general. In fact, all animals have a microbiome. Insects have a microbiome. There's a very intimate microbiome of turf nematodes and that sort of thing. I happen to have a patent that deals with adjusting the microbiome of mosquitoes and pests like that and turf nematodes, because there's a whole microbiome involved with all animals. If you look at the evolutionary history of those genes, they emerge in kind of coincidental with their host. So it starts getting you thinking, who's on which end of the leash? Have the bacteria steered the evolution of their host, or has the host just glammed on to the benefits of the bacteria? Are the bacteria just exploiting? They're farming us. We're just a field that they happen to attend to propagate themselves, or are we using the bacteria? It's an interesting evolutionary question, but all bacteria have a coevolution together. There's no such thing as the evolution of an animal and the evolution of a bacterium. They co-evolve to form meta-organisms. That's my kind of thesis. I think we're getting chased out, right? We're getting chased out. George, I hope I answered your question. Dean, I hope I answered your question a little bit anyway. Okay, you had some follow-up.
Yes, so is it able, is one able to find out whether you're having like regurgitation back into?
Yeah, you can get, you have a gastroenterologist?
Not here.
A gastroenterologist will do an endoscope and look at that and sample it and see if you have a bacterial overgrowth.